This is the Office of Research Integrity misconduct case about Logan Fulford. So he was a former graduate research assistant at Cincinnati Children's Hospital Medical Center, as well as a former graduate student at the University of Cincinnati. So he claimed to have found results that suggested some patients will express the transcription factor FOXF1, which is typically not expressed in a normal prostate. So basically, this suggested to him that FOXF1 increased prostate cancer by stimulating the kinases ERK5, MAP3K2, and WINK1. He also linked this overexpression to rapid tumor growth and metastasis in mice, and suggested that knocking down, using a knockdown route for therapeutic treatment of these tumors. He did this research through orthotopic transplantation models in mice and humans, in which he transplanted the FOXF1 transcription factor into tumor cells and then put these into the prostates of mice. He analyzed his data on immunohistochemistry, quantitative RT-PCR, various assay testing, as well as human patient data analysis. So his misconduct was image manipulation, in which he was found to be falsifying data in images which were in published articles. Well, it was in his published article in Science Signaling of 2016, and he also was found guilty of 3 accounts of fabrication that were in an unpublished manuscript. And in figure 2C, which you see on the right, he reused immunohistochemistry images. As we can see, the 2 in orange are exactly the same. They're just moved up and to the left a little bit. And as the blue ones, these are exactly the same images as well, just up and to the right. He also manipulated images found in supplemental data, which can be seen in figure S4E, as well as in figure 7C. These are both reused and relabeled Western blots. In the orange here, we see that there's different uses of exposures of lighting, and the same image was just relabeled. Here in blue, we see again alteration of lighting and increase or decrease in size. Here in the red, we see the use of backlighting and darkening, as well as rotations and size adjustments. Here in the green of figure 7C, we see differing exposures, which can be seen here. And then we also see some rotations as well as size scaling to make it seem different. Another manipulation of a Western blot, which was in figure S3B. Here in green, we see that there is splicing and resizing as well as rotation of the Western blot. So his sanctions, due to the misconducts that he performed, he required supervised research starting on May 8th, 2020, which will last for 2 years. He agreed to this responsibility and agreed to be in compliance with this plan of action. So for any further research that he wanted to perform, he required this plan of supervision that needed to be submitted and approved by the ORI. And this plan of action required at least 2 to 3 senior faculty oversight, in which they will review his primary data on a 6-month interval and just make sure that he is not falsifying any more data. And then any PHS applications, manuscripts, abstracts, just applications for funding, he will also need oversight for that as well. So the senior faculty will also be looking at that. He also voluntarily excluded himself from serving on any peer review, PHS, or advisory committees, as well as any boards, and they extracted his article on July 31st, 2018, and the article had been published on May 10th, 2016. So the impact of the misconduct. So for Fulford, his co-authors, and CCHMC, the institution that he worked for, they will ultimately be losing a lot of credibility from this. Fulford himself, he will face tons of obstacles when publishing. They will definitely be looking at his work with a little bit of a closer eye, as well as he might be losing out on some citations. Also, he's going to find struggling to when he's applying for funds just due to the lack of credibility that he has, and they're not going to want to give money to someone that they don't believe that they can trust. And as well as getting a new job, same thing. They're not going to want to give a job to somebody who has been seen to falsify data and not actually do the research that they claim to be doing. And as for the impact on public health, this government funding was ultimately wasted. It could have been used for someone who is actually doing true research in order to find a cure for prostate cancer or look farther into prostate cancer or any of those sorts. And here are the references. Thank you.